Okay, let's get things started. So, hello again, everyone. Welcome back. I hope you're all full and caffeinated and ready for the afternoon session. I'm Shahab Bakhtiari. I'm a professor, assistant professor in psychology department at University of Montreal, and I am happy to co-share the session with Federica this afternoon. So, very interesting set of talks with a theme. So you will see there's a theme. There's a very concrete theme to the talks, which I'm a fan of. I'm a bit biased, but still. Okay. So we can, the first talk is by Madineh, Dr. Madineh Sedigh-Sarvestani. She is an assistant professor at Cornell University in the Department of Neurobiology and Behavior, and at Howard Hughes Medical Institute Freeman Rabowski-Esclar. She earned a PhD in engineering science and mechanics from Penn State University and a BA from Harvey Mudd College. Her research examines how the architecture of visual circuits in the brain supports sensory processing, particularly how these circuits adapt to an animal's movements. Using techniques such as two-photon imaging and electrophysiology, her work spans multiple animal models, including treeshrews and mice, with the goal of advancing our understanding of embodied cognition and developing new neurological therapies. And today she's going to talk to us about comparative analysis of visual sensation and perception. And the floor is yours, Madineh. Thank you so much.
I guess I'll hold it. That seems like a bad idea. Yeah, let's do it that way. Thank you. Can you guys hear me? Doesn't sound like it. Better? Better? Right in my face, but that works. Okay, all right, let's get moving. Thank you. Okay, so this is going to be a two-part talk today. In the first part, I'm going to talk about my postdoc work, which I did with David Fitzpatrick at the Max Planck Institute in Florida. And it has to do with understanding representations of visual space in the brain. The second part is my relatively new research program at Cornell, and it has to do with sort of struggling to put the brain back into its embodied context. And like the graphics show, the first part is polished, it has productive value, it's been published. The second part is, in the works, and it's a new exploratory, but I think very exciting scientific adventure. So understanding representations or any sort of, neural code, the neural code in the brain, is complicated by the fact that, let's say you're working in visual cortex all the way in the back. The sensory organ, the retina, is all the way in the front, and there's a bunch of sort of wires and synapses that connect the front to the back. So there's a lot of, we keep hearing there's a lot of complex wiring and connectivity. But it turns out that within all of this complexity, a lot of quite clear organization emerges. And this includes the types of things you've all heard about. There's neurons in the visual cortex that prefer a particular spot in visual space, but not everywhere, a particular color, but not all colors. And one of the most sort of obvious organizational schemes is something called retinotopy. And it has to do with the fact that neurons that are close together physically on the brain, in the visual cortex and many other areas, also prefer small locations that are near each other on the retina or in the visual field, which gets projected onto your retina. So I'm going to show you what this looks like, and I think it'll be pretty obvious. So you're looking at brain activity recorded in the visual cortices left and right of an animal here through a cranial window, and it's a top view. And I'll play it again. As the awake animal is looking at this bar projected on the monitor, and therefore its retina. So I think what's going on is pretty clear. There's a corollary activity pattern on the brain that matches the visual stimulus on the sensory organ. And to make this relationship explicit, I'm going to use a simple color code, and I'm going to color each pixel on the brain with the position of the bar on the monitor that produced the most activity. This color map is known as the retinotopy map. You can see it right there. And these maps are sort of seen in many visual areas. And if you think about how you would sort of transform, the transformation between what's on the monitor, which is the same sort of approximately as what's on the retina, to the brain, it's pretty simple in this case. You take the monitor map, you put a grid on it, let's say, you flip it upside down, squeeze it a little bit, and you get the map on the cortex. And these maps that preserve local relationships and angles are formally known as conformal transformations, but for simplicity, I'm just going to call it a simple map because that's what it is. So I've only shown you one axis of the visual field. The visual field is this two-dimensional thing because the retina is two-dimensional. But for the other vertical axis, it's the same. It's a simple map. And this simple mapping is in some way comforting because it suggests that the patterns of activity on the brain have something to do with patterns of activity in the outside world. And this is sort of just comforting to us. So across all of these mammals in the evolutionary tree here, there's a big area in blue in the back of the head, V1, primary visual cortex, that has a simple map of space. The adjacent area, one synapse away, V2, shown in red, before our study was thought to also have, presumably, a simple map of visual space. We asked if this is true. We checked the rule in tree shrews, which I'll tell you about in a minute, and we found that in this other visual area, V2, this very prominent rule that could safely be relied on is broken. And I think many of you might not know what a tree shrew is, and I've become the evangelist for these critters. So allow me to make the introduction. Tree shrews are these little guys. They look and act like squirrels, except they're even more agile, if you can believe it. And they have a lot of advantages as models of the visual motor system. They're diurnal, unlike other small animals that can be studied in a visual circuits lab, which is rodents. Rodents are nocturnal. They have a smooth brain, which is perfect for imaging. So here you're looking at a head-fixed tree shrew, and this is a brain window, a cranial window with wide-field calcium imaging, and that's zoomed in with 2P calcium imaging. So this means that viral vectors largely work out of the box, even though they were designed for mice. They work in tree shrews. They have a short gestation period, so compared to marmosets, other small actual primates, they have that advantage. But yeah, every model organism has its own advantages and disadvantages, and species diversity is good. So tree shrews are just sort of the animal I ended up with. And we use this setup, head-fixed calcium imaging in awake animals, to look at the retinotopy maps in V1 and V2. And we expected the maps in V2 to be simple like the maps in V1. So something like that. And this is published work, so I'm going to skip sort of a bunch of the difficult data, but if you're interested, you can look it up. What we found in V2 of tree shrews was this sort of stripy, complicated map. And it took a while for us to understand it, but basically, the question is, how do you get the transform from the monitor, or from V1, which is how V2 gets its inputs from the retina? How do you transform those inputs to go from the simple sort of continuous linear map on the monitor to the striped map in V2? And you can do an exercise again. You put a grid on the monitor, and you have to actually kind of stretch the grid so that it's no longer two-dimensional, it's one-dimensional like a rope. And then you fold that rope over in on itself, and you get these stripes. So this is a really different representation of visual space where the actual transform is something like this back and forth, snaky, twisted, we call it sinusoidal pattern that connects the retina to V2, or in this case, V1 to V2. So this is along one axis, and it's basically if you take every point in this thing, let's say V1, and you transform it using this pattern, you end up with this other pattern in V2. And along the other axis, the vertical dimension of retinal space, you're using the same transform, but now it's moving along the gradient, so you would get sort of this continuous map in the second dimension of space. So to summarize this little bit, unlike every other map reported in the literature, where cortex has more or less a simple transform, a simple, continuous, linear-like transform of the retina that preserves its layout, you can see the retinal image still in these simple maps in V1 and other areas. The maps in V2 combine the two axes, the horizontal and vertical axes of visual field, into essentially a single long axis so that you no longer preserve the retinal image. You've sort of folded it in on itself. And so the functional effect of this is that points that are close together in space on the retina are still close together in V2, but also points that are a little bit further apart, especially along this horizontal axis, are now also close together on the brain because you've made this sort of twisted transform. So when we saw this, and it took a little while to figure it out, but we thought, okay, well, this has to be a weird tree shrew thing. They're weird animals. This hasn't been reported. It can't generalize. And it turns out, and we have a lot of evidence, and again, this is published, that it does generalize. So we measured the same maps in V2, or more properly, area 18 and 19, it's called, in ferrets. And we saw very much the same striped pattern in both the functionally measured maps of visual space and their anatomical correlates, which I haven't really mentioned until now. But if you look at the connections between the left and right visual cortices, the central portions, the neurons that represent sort of the line that divides the two hemifields, they send projections to each other. And the idea is that helps to sort of smooth the hemifield representations across your two hemispheres. Those anatomical projections carry a signature of the striped or sinusoidal transform, and we can see that signature in ferrets and also in macaques. And so we suspect that if you could have high-resolution calcium imaging in primates, maybe marmosets that also have a smooth brain, you would see this same retinotopic mapping, and there's a little bit of evidence from heroic electrophysiology experiments where you go in. I mean, V2 in primates is difficult to measure because it's stuck in a fold, but you can go in with an electrode just at the right angle, and you can write down the position of the receptive field, the preferred location in visual space, as you go along sort of this long axis of V2. And when you do that, you get this sort of discontinuous ratchet pattern that I think, given our observations, is probably a low-sampled version of the sinusoidal transform. Okay, so that's the pattern. That's sort of the what. How's this set up? We sort of land back at the question of you have these simple maps, these simple representations that basically kind of mirror the retina in V1. How do you get this complex map in V2? There's sort of one possible answer. The simplest answer is that the synapse between V1 and V2 is the sinusoidal transform. And that's the simplest possible answer because V2 gets most of its inputs from the retina through V1. So to test that, we use colored tracer injections. So I'll walk you through this. You're looking at a top view of a flattened section of the tree shrew brain that contains a little bit of V1 and a little bit of V2. And we've injected small amounts of a fluorescent dye that goes into the cell body, and so you can see the axon patterns. And this isn't real data. This is fake data that I've mocked up to show what we would expect if the maps in V2 were simple. You would basically expect this is a new visual area. You would expect a mirror one-to-one mapping. of what you injected in V1 and V2. Because the maps should be the same, maybe they just reflect each other. What we actually saw is something like that. So for each point, there's a couple of interesting things here. For each point in V1, you're getting multiple stripes in V2. The stripes have gaps between them, and the color order flips. Here you go from orange to green, there's a gap, and then you go green to orange, and again. And so these holes in between are places in V1 where you don't have label. And if you sort of do a mental model, you sort of draw that sinusoidal curve across these injections, you get exactly this pattern. So it is this very complex, but sort of mathematically well-defined synaptic connectivity between these two areas that gives us this complex representation of visual space in V2. And I want to draw your attention to an important point here, which has to do with the color reversals that I mentioned. When you get these color reversals, in the mouse literature, these reversals are thought to be reversals in progression of the visual field, because here, this is a smooth map, so as you go across these colors, you have a progression of the visual field. So when you get these reversals in the axons, it's the mouse anatomists draw basically areal boundaries every time the color order flips. And that's how we end up with something like 16 higher order visual areas in the mouse visual cortex. That's all under the assumption of sort of simple retinotopy. So my point here is, now that we know that retinotopy in these, relatively low visual areas like V2 can be nonlinear and complex and fold in on itself, these areal boundaries need to be revisited. Okay, so how much time do I have? Okay, great. So that's the what and the how. The coolest question is why? Why does V2 have this weird representation? Why questions are fun and, of course, really hard to answer, because to really get at this question, we'd probably need evolutionary perturbations that we can't do, maybe are impossible to do. But I like following these types of questions anyway, because they help sort of design experiments and inject some fun experimental juice into your boring day. So go along with me. So one answer to why, and again, why would V2, the stretched out long area, have this weird representation? The sort of throwaway answer, and we have a series of simulations in the published work, is, okay, right, well, this area is very elongated in all these animals, and the optimal representation of the visual field, given the cortical constraints, is this representation. Sure, but that's sort of just a chicken and egg. That throws the bucket to, well, why is V2 elongated in the first place? So I think a better answer is that this representation might have something to do with energy efficiency in the brain. And most of our answers to why questions are always energy efficiency, right? We kind of have one answer, efficiency. But let's go with that answer now. So to show you what I mean, how would this representation be sort of make anything, any computation that's relevant to the animal more efficient? I'm going to use an example called the log polar transform that's been observed in actually V1 of certain primate species. So it's a conformal transform in that it preserves local relations in the retinal image, but it has additional nice properties that make it an efficient transform. And they have to do with the fact that this log polar transform converts polar angle and radius on the retina into horizontal and vertical dimensions on the cortex. So you sort of do this coordinate transformation. And this means that if the retinal input is rotating, which requires keeping track of 2 dimensions, the equivalent cortical activity is just sort of a linear wave, which we know cortices seem to do easily. And if the retinal image is something that's expanding, the equivalent cortical activity is a linear wave in the orthogonal dimension. And there's nothing special about a bar rotating or something expanding, but of course these are retinal images that can be and are often produced by our own movements. You move towards something, it expands in your field of view, you tilt your head, you get rotations in the visual field. And now it should be easier why this log polar transform is useful and why it's used in robotics and image registration all the time. So can we tell a similar story with the sinusoidal transform in V2? In V2, the pattern of input that would produce similar wave-like cortical activity looks like this. It's the snaky pattern. That's how you get activity from one side of V2 into the other. And is there a pattern of body movements, or like maybe a fly in the tree shrew ecological niche that moves in that way? I couldn't really think of anything external that moves in that way, but tree shrews do constantly make these head movements that are sort of like primate saccades in that they're used to sample the visual environment. So maybe that's why. Maybe the animals move their heads like this, and so if V2 has this transform that folds space so that relatively far, like things that are 30 degrees apart in the visual field are close together in the brain, that helps the animal sort of have smooth vision across these head saccades or do foreground-background subtraction. But I think this is a really throwaway and unscientific answer because I don't know the behavior of these animals at all. I don't know how often they make these head saccades. I don't know if they're actually relevant in the way that I'm telling this story. And that's because most of our understanding of the tree shrew visual cortex, which is actually significant, comes from animals that have been specifically constrained so that they don't move. And that's not just true of tree shrews. It's true of most small lab animals like mice and rats too. Visual neuroscientists, and I don't mean all visual neuroscientists. Here I'm focusing on my little subfield, which is circuit neuroscientists, have by and large ignored body movements in favor of measuring brain activity using things like 2-photon microscopes. So we have a limited understanding of the visual system in its natural embodied context. So at this point in my postdoc, I independently arrived at an argument that many other people, including Paul Chizek and others in the room, have been making for years, which is that the brain and body evolved, function, and malfunction together, so we have to study them together. And I realized that what I had been studying was sensation, when what I was really getting more and more interested in, especially in tree shrews that, as you'll see, are really made to move, was perception. And I need to distinguish these two terms, I think, even for this crowd. Sensation is the activity in your brain that results from inputs to your sensory organs, but it doesn't have meaning. And so it's inactionable. For instance, if I said Danishgah versus Dabiristan, you would hear these sounds as different. Your auditory cortex would receive them as different. But unless you speak Farsi, and actually this does not work in this crowd, Salam hamvatanan, in most crowds, unless you speak Farsi, which most people don't, it sounds like visual gibberish to you. It doesn't make sense. You would need to be a native speaker, or you would need to have a dictionary to translate the sensation into perception in order to know whether I said university or high school. Perception, on the other hand, is what happens when you have this dictionary and you use that to make simple associations of certain sensations with certain contents, and you get perception that is grounded, actionable, and it has meaning. And believe it or not, vision is the same way. It seems that when we look, we are sensing and perceiving at the same time, because it's really hard to imagine visual gibberish. It's hard to imagine something that you can see, like a shape or a color that you see but you don't recognize. But vision works the same way as language. You have to learn it. You have to learn to make associations that add meaning to sensation over a period of weeks. And we know this in part from people who we give sight to suddenly. So people who get their cataracts removed in adulthood, they have difficulty immediately perceiving, immediately recognizing a face as actually a face it takes them a little time to sort of experience the world with this new sensory ability. So the hypothesis here, and it's not my hypothesis, but it's one that I'm liking more and more, is that this dictionary, to translate visual sensations into perceptions, is rooted in body movements. In other words, body movements are what gives meaning to sensory gibberish. So I call this the sensory motor dictionary hypothesis. And I'm going to give you an example that should hopefully make it a little less abstract. Say you're looking at this bust or statue that's directly in front of you, and you look to the right, and the position of the thing, of the bust, on your retina changes in a systematic, predictable way. And you look to the left, and it changes again in the opposite but systematic and predictable way. And this sensory motor pattern, this combination of changes on your retina and the eye movements that you made together define that something is directly in front of you. So the sensory motor input is the definition, dictionary definition of something in front of you, and only when something is in front of you and you get this exact sensory motor pattern, so there's this one-to-one mapping that has this precise sensory motor signature or precise sensory motor definition. And we can do this for other properties of the physical world. So let's say there's this horizontal branch and you're a monkey or a tree shrew. The pattern, if you look right at it, the pattern on your retina is a horizontal thing. If you now move your eyes or head against it, the image on your retina doesn't change very much because horizontal things are self-similar under lateral sensor movements. But if you moved your eyes or head up or down, the retinal image will shift drastically, again, in a systematic, predictable way. So this sensory motor pattern can define whether something is horizontal or vertical. So the whole idea is that body movements coupled with predictable reafference, predictable change in your sensory input, can define properties of the physical world that are actionable and immediately meaningful. And once you grasp this concept, the experiments are pretty clear, and they're rooted in sort of computationalism and input-output frameworks that we already use. Are there neurons in the brain that are tuned to these sensory motor patterns? And can we show that brains learn these definitions through experience with bodies? Those are the two main big questions. And they're hard questions because we need to measure a whole bunch of things. We need to measure body movements, the coincident change in visual inputs and probably vestibular inputs, and brain activity, all in the same freely behaving animal. And this is a ton of work requiring technology that's still largely developing. But I felt like for this animal model in general, I didn't have a choice because so far most of circuit neuroscience has focused on sensation, visual gibberish. And we have learned a lot from this process. This conference, the deep learning revolution is very much contingent on this type of work, and we need more of it. But I think some subset of us circuit neuroscientists working on these small lab animals where you can make all of these measurements need to pay attention to perception. And I think if we did that, we could design better therapies for blindness. We could maybe understand hallucinations and dreaming, actual visual gibberish, or maybe not. And maybe we can make better self-driving cars. I was promised a self-driving car years ago, and it's still sort of not quite there. And the same goes for neuro AI. Let me make a point and attach myself to neuro AI. The same goes for neuro AI, as we were talking about yesterday. I know that if we give ChatGPT a body, it will be able to hurt us more and in worse ways. But I think until AI is embodied, we are going to be limited to sensation. And I think that we can't just have virtual movements until we get these sort of measurements in real animals, model them, understand them, and then we can make virtual embodied AI. But for a while, we need robots with bodies. So here's a snapshot of our progress in the last 6 months since my lab was built. This here is a tree shrew wearing a GoPro camera. We use deep learning models to quantify their natural-ish movements. That's the motor part, and the GoPro camera obviously is the visual part that gives us the sensory motor definition. And you might have already realized that to really fully test this dictionary hypothesis, you need at least 2 animals that move differently. And that would allow us to test this idea that the more complex an animal's movement repertoire, the more complex, the more thick its sensory motor dictionary, and therefore the more complex its perceptual experience of the world, the set of possible perceptual experiences. So the second animal you saw there, if you were paying attention, was a rat. And here's another video, if it plays, that shows you just how different these 2 animals' movement patterns are. And here we're imposing the same exact optics because it's the same little camera on the head. The visual environment is largely, again, imposed to be the same. So the factor that is isolated here is the movements and how they influence the sensory motor input patterns that define the dictionary. And one thing we can do here is sort of try to understand this complicated dictionary, but the other much more simple thing is that we can just study the statistics of sensory motor inputs, which will be very different just because the rats and shrews move really differently. And so this is a collaboration with Shahab Bakhtiari, and I'm really excited about the shape that it will hopefully take in the next couple of years. 2 minutes. So yeah, we've started to quantify the movement repertoire of these animals sort of in a serious way using another class of deep learning models that people like Eva Dyer, who will be here tomorrow, and Bob Dodd at Harvard have developed and made publicly available. And to summarize this briefly, the models can recapitulate something very obvious from the video, which is that tree shrews have a more complex movement repertoire. Okay. And this sort of analysis allows us to predict based on, this is sort of the end goal, based on the movements, we should be able to predict what physical attributes of the environment tree shrews should be better or faster at perceiving compared to rats. That would be a cool question to ask. And this is a prediction that we can then test behaviorally. And I just want to give a shout out to the many other folks, especially in the human vision world, that have been doing this kind of work for years. Most notably, Mary Hayco at UT Austin has been making really precise measurements of body movements in humans doing normal activities like hiking or crossing the street, eye movements using that head-mounted device, and that allows Mary's team to then estimate what people are seeing in a really precise way, and she's already sort of already able to quantify that this information, the sensory feedback, is really rich. So for instance, if you look at this flow and somebody crossing the street, both its speed and its direction can separate motion on the retina due to self versus others, which is sort of a favorite problem that visual neuroscientists always talk about. How does the brain know if I'm moving versus something else is? Well, it's right there in the data. The brain doesn't have to build a model. Okay, and I think I'll stop there. Thank you.
Thank you so much for the great talk. Super interesting. We have time for maybe 2 relatively short questions. Paul.
So first of all, great talk, and thanks for citing me. That was nice. I have a question actually about the maps that you find. Are they very consistent across animals? And have you looked at I know this is asking a lot, but have you looked at where how their projections go further downstream? So do they remain segregated in some way and connect differently, sort of dorsally versus ventrally versus different somatosensory maps?
Yes, the first question, we looked in ferrets, they're definitely there. Anatomical evidence suggests that they're there. Also in squirrels, in cats, and in primates.
Oh, sorry, I meant across individual tree shrews. Very consistent, okay.
Thanks. Second point, I'd love to do that. So it would require careful injections into the different stripes and essentially defining the 2 pathways in the tree shrew visual system. We, and no one else has done that yet, but we would love to.
Well, so because I'm wondering, because the somatosensory maps also have this mirror reversal pattern, right, in mammals, and so I'm wondering whether there's a correspondence between them, when there's sort of the gradients are aligned in the
I've thought along those
visual.
lines. I think this is a self-organizing scheme that's present elsewhere too.
Hi, super interesting talk. Thanks. I follow like your sensory motor connection for like the location of certain objects and so on, but like actually the identity, like it's harder for me to follow why like moving my head would be very important to recognize your face.
Yeah.
Like, what's your thoughts on that?
My thoughts are that sort of understanding the amount of information about the physical world and our own bodies that is present in sensory feedback requires understanding geometry, physics, and just having imagination. And I would recommend the book Action and Perception by Albano, where he really sort of gives a lot of other examples. And, you know, this guy's a philosopher. And I think we need to bring that sort of thinking into neuroscience and build testable hypotheses around it. Thanks.
Okay, thanks.